of microbiology, we are going to discuss about antigen in full detail. Okay, watch the video full for getting all the informations about antigen. Clear? So first we will define antigen. Antigen is any substance that satisfy two immunological properties. The first one, it have antigenicity and it must have immunogenicity. Okay, so any substance having these two properties will be considered as antigen. Clear? Now we will discuss what is immunogenicity and what is antigenicity. So first coming to the immunogenicity. What is immunogenicity? Okay, so immunogenicity is the ability of antigen to induce immune responses in the body. Okay, immune responses in the body. So immunogenicity is the ability of any antigen to induce immune response in the body. Clear? One example I will take here. Suppose antigen comes and stimulate B cells. Okay, and convert B cell into, we know conversion of B cell into effector B cells plus memory B cell. Effector B cell is also known as plasma B cells. Okay, so this antigen is said to be have immunogenicity because this antigen is inducing B cells to convert into effector B cells and memory B cells. Clear? Now coming to the antigenicity. So it is the ability of an antigen to combine specifically with the final products of the above reactions. Means suppose if antibodies are produced by plasma cells, then with this antibody, antigen must bind. If antigen is binding with these antibodies or other products, then it have antigenicity. So antigen must have this property, that is antigenicity as well as immunogenicity. Clear? The substance that have only immunogenicity are called as immunogen, not antigen. Okay, that substance is only called immunogen because this is only have immunogenicity. Clear? So this is the basic difference between antigen and immunogen, antigenicity and immunogenicity. Now we will discuss about one important term here, that is known as epitope. Clear? So epitope, it is also known as antigen determinant site. Okay, epitope is known as antigen determinant site. Okay, and it is smallest unit for antigenicity. Clear? Now what is epitope again? So what is the basic definition of epitope? How we will define? So it is defined as a small area present on the antigen, comprises few, that is 4 to 5 amino acids or monosaccharide residues. Okay, that may be amino acid, that may be monosaccharide residues, and it is capable of sensitizing T and B cells. Okay, with a specific site of T cell receptor or an antibody. So once again, epitope is a small area which is present on antigen, comprises of few amino acids or monosaccharide residue that is capable of sensitizing T and B cells and reacting with a specific site of T cell receptor or an antibody. Clear? So this is your epitope. Epitope is present on antigen, and this one term more, that is paratope, it is present on antibody. So this is the basic difference between epitope and paratope. Now this epitope is grouped into two categories. Okay, there is two types of epitopes. The first is your sequential or sometimes also known as linear epitope. The second one is your conformational or sometimes also known as non-sequential. So this type of epitopes, it has only single linear sequence of few amino acids. Okay, so it has single linear sequence of few amino acid residues, whereas this conformational or non-sequential, it is formed by bringing together the surface residues of different sites of peptide chains. Okay, different sites of peptide chain. So this is formed by bringing together the surface residue from different sites of peptide chains. This is by only one peptide chain. So basically you can differentiate that it involves only one peptide chain, whether it involves many peptide chains. So okay, this is your epitope. Now moving to the next important points, that is your heptane. One important point, heptane. So heptanes are those molecules which are antigenic in nature. This is antigenic in nature, but they are not immunogenic. Heptanes are not immunogenic, they have antigenicity. Clear? So what are heptanes? So heptanes are low molecular weight molecules that lack immunogenicity but have antigenicity. Clear? Now heptanes can become immunogenic when it combines with a large protein molecule called as carrier. So when it hapten combined with carrier protein, then it will be immunogenic because its size will increase and it is able to have properties like immunogenicity. Clear? Now the hapten carrier complex which is formed, okay, after combining with this protein, so hapten carrier complex is formed and it is capable of inducing immune responses in the body. Okay, it is able to cause immune responses, and it is observed that animals which are immunized with such a hapten carrier conjugate produces antibodies which are specific for, it is specific for either epitopes of hapten, okay, that antibody which are produced maybe for the epitopes of hapten or epitopes of the conjugate protein which you have placed, epitopes of conjugate protein, or those antibody may be for new epitope. What is a new epitope? So new epitope, it is may formed by combined path of both the hapten and carrier protein. Okay, by the combination of hapten or carrier protein, there is chances that new epitope may be formed. Okay, so antibodies are also produced against that new epitopes. So antibodies are produced against the epitope of hapten, epitopes of conjugate protein, and new epitope which are formed by the combination of both. Clear? Now hapten has been classified into two types again, that is the complex haptens and simple haptens. So these simple haptens usually contain only one epitopes. Clear? And these complex haptens contain 2 or 3 epitopes. So they can react with a specific antibody, and the hapten antibody complex can be visualized by various methods such as the precipitation reaction. Okay, and these simple haptens can bind to the antibodies, but the hapten antibody complex cannot be visualized. One important point, this is not visualized. Okay, simple will not be visualized, complex will be visualized because it will give precipitation reaction because its size is more, but simple size is less because it has only one epitope, so it will react with only one antibody, so precipitation is very less in simple type of hapten. Clear? So this is all about your hapten. So we have discussed antigen and hapten molecules. Now we will discuss about antigen host relationship. Clear? So based on the antigen host relationship, antigens can be grouped into two groups as okay. So the first group is self antigen, which is also known as autoantigens. Clear? So they belong to the host itself, so they are not immunogenic. So they are not immunogenic to the host because this antigen, the self antigens, are the antigens which belong to host itself, so they are not immunogenic in nature. Clear? Host immune system does not react to its own antigen, which is due to exhibiting a mechanism which is known as immunological tolerance, which we will discuss in a different video. What is immunological tolerance? It is because of that immunological tolerance host antigen is not responding, not immunogenic. Clear? However, in certain conditions, self antigens are biologically altered, as in cancer cells, and can become immunogenic, but that is in some disorders, not usual. Now, second classification is non-self antigen or foreign antigen. It is also divided again into 3 types. Okay, the first one is alloantigens, the second one is isoantigens, and the third one is heteroantigens. Clear? And non-self or foreign antigens are immunogenic. They are immunogenic because they are foreign, so they are immunogenic. Now coming to the first, that is alloantigens. So these antigens are species specific. Alloantigens are species specific. Clear? And tissues of all individual in a species, contains these species specific antigens. Clear? Now one more point, these alloantigens are immunogenic in nature. These are immunogenic in nature. Clear? Now the next is your isoantigens. So these are the types of antigen which are present in only in subsets of species. Okay, only in subsets of a species, such as blood group antigen and histocompatibility antigens. Okay, so these are coming under the isoantigens. Clear? Now the next one is your heteroantigens. So heteroantigens contains antigen which is belonging to two different species are known as heteroantigens, as hetero word is saying. Okay, so two antigen which is belonging to two different species are called as heteroantigens, such as antigen of plant or animal or microorganism. These type of antigens are included under this. Clear? Now one more point, that I need to mention, self antigen are not immunogenic. Clear point. Whereas heteroantigens, these heteroantigens and these alloantigens are immunogenic. Okay, the degree of immunogenicity increases with the distance. We will discuss later on. Now coming to the isoantigens. Isoantigens, these are antigens which are not immunogenic to those individuals who possess these antigens, such as blood group. Okay, you are knowing the antigen which is present on the RBC of that particular species, that particular person. Okay, are not immunogenic, but different antigens in such as blood group of different person. Okay, that do not matches with the particular person. Okay, and if that blood group is coming inside this human, okay, have different antigen, then antibody will form against those antigen, but not against these antigens. Clear? So isoantigens are not immunogenic to those individuals who possess this antigen, but for other individuals they are immunogenic. Clear? So antigen host relationship is over. Now we will discuss about one more point here, that is heterophile antigen. This is important concept. Okay, heterophile antigens are a type of heteroantigens. We have discussed heteroantigens. So this is a type of heteroantigens. Clear? That are present in two different. Heteroantigens means present in two different species. Heteroantigens means antigen which are presenting in two different species. Clear? So heterophile is a type of heteroantigen. Clear? One important point, what is the unique inside the heterophile antigen? So in heterophile antigen, they share epitopes with each other. They share epitopes with each other. Suppose this is species A, this is species B. Okay, it has antigen, it has antigen. Those are different species. Okay, A species, B species, but its antigen and its antigen, both antigen will share epitopes. So if antibody is forming inside this antigen, then this antibody can be used for this also. Clear? So antibody produced against antigen of one species can react with the other and vice versa. Getting? Now what is the diagnostic application of this heterophile antigen? That is very important. Diagnostic application of that antigen, heterophile antigens. So the first is Weil-Felix reaction. The first diagnostic application is Weil-Felix reaction. So it is basically done for typhus fever. Okay, Weil-Felix reaction, that is done for typhus fever. In this, what we will do, antibody which is against rickettsial antigens are detected by using cross reacting with protease antigens. Okay, so antibody which are formed against your rickettsial antigens are detected by cross reacting it with protease antigen, because protease antigen as well as rickettsial antigen both share similar epitopes. So antibody which are produced against this will also work for this. Clear? So this is Weil-Felix reaction. This is very important. Weil-Felix reaction is associated with typhus fever. The second one is Paul-Bunel test. Paul-Bunel test. So this is done for infectious mononucleosis which is caused by Epstein-Barr virus, A virus. Okay, so Paul-Bunel test is done for infectious mononucleosis which is caused by Epstein-Barr virus. Okay, in this sheep RBC antigens, okay, sheep RBC antigens are used to detect cross-reacting antibodies which are present in patient sera. Clear? So Paul-Bunel test is associated with Epstein-Barr virus, whereas Weil-Felix, okay, Weil-Felix reaction is associated with typhus fever. Now the third one is your cold agglutination test. Okay, this test, cold agglutination test, as well as one more test I would like to include here, Streptococcus MG test. Cold agglutination test and Streptococcus MG test are done for primarily atypical pneumonia. So cold agglutination test as well as Streptococcus MG test are done for pneumonia. Okay, here antibodies which are forming against Mycoplasma pneumoniae are detected by using human O blood group RBC. human O blood group, RBC, and Streptococcus MG antigens. Once again I am repeating, here antibodies which are formed against Mycoplasma pneumoniae are detected by using human O blood group, RBC, and Streptococcus MG antigens respectively. Clear? So this is about your cold agglutination test. So we have discussed 3 tests. Once again revising: Weil-Felix reaction, then we have discussed Paul-Burnell test, okay, WF, PB, and then cold agglutination test and Streptococcus MG test. So this is about your antigen host relationship. Now we will summarize the factors which are affecting immunogenicity. So the first factor is size of antigen. Okay, larger is the size, more potent is the molecule as an immunogen. The second one is your chemical nature. Okay, I need to mention here proteins are the stronger immunogens than carbohydrate, which is followed by lipids and nucleic acid. So proteins first, then carbohydrate, then lipid, then nucleic acid. The third one is susceptibility of antigen to tissue enzymes. Okay, molecules that are not susceptible to tissue enzymes, such as your polystyrene latex or synthetic polypeptides composed of D amino acids, are not antigenic, while polypeptides which consist of L amino acids are antigenic as they are degraded by tissue enzymes. So tissue enzymes also play an important role. Remember, from PG point of view, D amino acids containing will be not antigenic, L amino acid containing will be antigenic. Now the next one is structural complexity. Okay, simple homopolymers made up of single amino acid lack immunogenicity, while as polymers made up of 2 or more amino acids are immunogenic. Clear? So more the structural complexity, more the immunogenicity. So the first factor is size, then chemical nature of the dose, then tissue enzymes, then structural complexity. The next one is foreignness to host. Foreignness to host. Okay, this is very important characteristic. Higher is the phylogenetic distance between the antigen and the host, more is the immunogenicity. Clear? Now the next one is your genetic factors. So on the basis of genetic factors, we have divided into 3 groups: responders, slow responders, and non-responders. Responders are those individuals who produce antibody faster, slow individuals which slowly produce antibody, non-responders who do not produce antibody. Clear? Then the next factor is your dose of antigen. Okay, antigen is immunologically active only in optimal dose. Okay, the next factor is very important, route of antigen administration. So important from MCQ point of view, route of antigen administration is also important. How? So see, IgA, okay, immunoglobulin A are better induced following oral administration of antigens. Clear? Inhalation of pollen grains, antigen produce IgE synthesis. IgE synthesis when there is inhalation of pollen grain. When the same antigen giving through parenterally route, okay, will induce IgG antibody. So pollen grain through inhalation produce IgE, through parenteral route IgG. IgA will be produced when there is oral administration of any antibody. Clear? Now site of injection also matter. Okay, hepatitis B vaccine. Hepatitis B vaccine is more immunogenic following deltoid injection, okay, in compared to gluteal injection. Hepatitis B vaccine is more immunogenic following deltoid injection than gluteal injection. Clear? So these are the factors which is affecting the immunogenicity: size, chemical nature, tissue enzymes, structural complexity, foreignness to the host, genetic factor, dose, route. Clear? Now we will move for the next part, that is biological classification of the antigens. Clear? So this is a video which contains all the important points about antigen. I will not leave any important point. Okay, so depending on the mechanism of inducing antibody formation, antigens are classified as 3 types. The first one is your T dependent antigens, the second one is T independent antigens, and the third one is your super antigens. Okay, how we have divided depending on the mechanism of inducing antibody formation. Clear? How they are inducing antibody formation. So first we will discuss about TD antigens, that is T dependent antigens. So most of the normal antigens are T dependent antigens. So most of them are T dependent antigens. Clear? And we know that antigens are processed and presented by antigen presenting cell. Okay, antigen presenting cell will present that antigen to T cell, which lead to T cell activation. Okay, and the activated T cell will release certain cytokines that in turn stimulate B cell, and this B cell will start forming antibody. Once again repeating, T cell dependent antigens means antigen presenting cell will present this antigen to T cell, then T cell will be activated, release certain cytokines which will activate B cell to leading production of antibody. So it is T cell dependent antibody production. Now the next one is TI antigens. TI antigens means it will produce antibody independent of T cells. How? So there are few antigens such as bacterial capsule, flagella, lipopolysaccharides that do not help the need of T cells and antigen presenting cell. They directly bind to immunoglobulin receptors. They directly bind to immunoglobulin receptors which are present on B cells, okay, and stimulate B cells. Clear? There will be stimulation of B cells. So bacterial capsule, flagella, lipopolysaccharide, they directly bound to antigen receptors which are present on B cell and stimulate B cells to produce antibody. Clear? And it leads to increased secretion of non-specific antigen, non-specific antibodies. Okay, specificity lower here. There will be secretion of different types of antibodies here. Clear? Now the next point, that TI antigens can activate both mature and immature B cells. Clear? One more important point where I need to mention here by changing the color, that in T cell independent antigens there will be no formation of memory B cell formation. Okay, mind it, very important point. So I have mentioned like this: in T cell independent antigens there will be no formation of memory B cell. Memory B cell will be not formed here. Clear? One more point, that in this type of antigens activated B cells do not undergo affinity maturation, class switchover, which we will discuss in different video. Clear? Here B cell will not undergo affinity maturation and class switchover. These are 2 points you must remember. I will discuss in different video, okay, about this in detail. Clear? Such an activated B cell can produce only limited class of antigens such as IgM and IgG3. So this type of B cells which are not undergoing affinity maturation and class switchover will only produce IgM and IgG3 type of antibody. Okay, once again I am repeating, this type of antigens will not involve formation of memory B cell, as well as only limited to IgM and IgG3. Clear? Now the third is very important, that is super antigens. So super antigens are the third variety of biological class of antigens. Okay, the unique feature of the super antigens is that they can activate T cells directly. Okay, they can activate T cell directly without use of antigen presenting cell. In T dependent antigens we have first use APC, antigen presenting cell, then we have activated T cells. In T independent there is no activation of T cells, but in super antigens there will be activation of T cells, but directly without use of antigen presenting cells. Clear? How it is occurring? So it is occurring by different methods, different mechanism. So the variable B region of T cell receptor, which is variable B region of T cell receptors, appears to be the receptors of the super antigens. So it appears at receptor for super antigens, means super antigens will come and directly bind to the the variable B region of T cell receptors. Clear? They directly bridge non-specifically between MHC2 of antigen presenting cell and T cells. Okay, these antigens directly form bridges between MHC2 type of cells and T cells. Clear? They directly forming bridges, non-specifically forming bridges. Getting? Like this. Like this. Suppose this is antigen presenting cell, okay, and it is binding with your T cell, then antigen will come and form a bridge between these two. Okay, this is the second mechanism. Now the third mechanism, that there will be non-specific activation of T cells, okay, leading to massive release of cytokines that can activate another T cells. Okay, so cytokines from activated T cell will stimulate other T cells to produce antibody. Clear? Now some examples of the super antigens. So the most important is your staphylococcal as well as streptococcal antigens. They are the examples of superantigens. Clear? They are very important examples of superantigens. And superantigens, one more point I need to mention is superantigens when stimulated led to hyper gamma globulinemia. Why? Because this is the non-specific activation. Again, cytokines which are released from activated will lead to release of more and more cytokines. Activate another T cells, will activate B cells and lead to more formation of antibody because of these reasons. Clear? So this is about your superantigens. There are some diseases which are associated with superantigens. I need to mention the toxic shock syndrome, food poisoning, skeleton skin syndrome, like this type of disease are associated with this. Clear? So this is all about your antigens.